After studying this module, you shall be able to know the crucial role played by iron in various metabolic processes, learn the significance of iron homeostasis, that is why it is important to maintain a constant balance between iron uptake, transport and storage, identify the iron transport and storage proteins Transferrin and Ferritin respectively, study the structure, function and mode of action of Transferrin, study the structure, function and magnetic properties of Ferritin and how this protein stores and releases iron, acquire information about iron related disorders such as iron overload, namely hereditary hemochromatosis, transfusional iron overload and iron deficiency anemia. Iron is a vital element that is required by all forms of life on earth as it serves as an integrated part of many important enzymes that play a crucial role in several metabolic processes such as oxygen transport, deoxyribonucleic synthesis, DNA synthesis and electron transport. Since iron is required for a number of diverse cellular functions, therefore it becomes important to maintain a constant balance, that is iron homeostasis, between iron uptake, transport, storage and utilization processes. During the past decade, advances in our knowledge and understanding of the structure and function of the iron storage and transport proteins, namely transferrin and ferritin, have greatly accelerated. The following sections elaborate the specific role played by each of these metalloproteins. So firstly, let us understand how iron transport occurs with the help of the iron transport protein called transferrin. What are transferrins? Transferrins are a group of non-heme iron binding glycoproteins that can effectively control the level of free iron in biological fluids. Transferrin glycoproteins bind tightly to iron but reversibly in the cleft of each of the two homologous lobes and hence capable of transporting it to the sites of absorption, utilization and storage. They are widely distributed in the physiological fluids of most vertebrates as well as invertebrates, including serum, egg white, mammalian milk, tears and leukocytes. These transferrin families are customarily divided into the following major categories based on amino acid sequence, function and source from which each is isolated. Firstly, serum transferrin. Serum transferrin, also called as serotransferrin, siderophilin and 13A metal binding globulin, is generally present in the serum of vertebrates and some other biological fluids. It plays a vital role in the transport of iron within the circulatory system of vertebrates between different biological tissues. Secondly, lactotransferrin. Lactotransferrin, also termed as lactoferrin, is generated by mucosal epithelial cells of mammals and therefore it is found abundantly in mammalian milk. In addition, it is also present in specific granules of polymorphonuclear leukocytes and other secreted fluids such as tears, pancreatic juice, etc. It plays a pivotal role in body defense against infections owing to numerous outstanding properties like antioxidant, anti-inflammatory and antimicrobial activities. Third, ova-transferrin, which is also called as conalbumin. Ova-transferrin or conalbumin, which is commonly found in oviduct secretions, eggs of birds and reptiles, possess antimicrobial property that is vital for birds' innate immunity. The structural protein of ova-transferrin is similar to that of serum transferrin since they are derived from the same gene. Fourthly, melanotransferrin, which is also a P97 cell surface protein. Melanotransferrin is one of the first cell surface markers associated with melanomas. The exact biological function of melanotransferrin is unknown. However, it has been predicted that it may play a major role in cell proliferation, migration, angiogenesis and tumorigenesis. What is the role of transferrin? Transferrin is mainly produced by the liver cells, which is then secreted into plasma. The main function of transferrin is to transport iron from absorption centers to the sites of utilization, storage and hemoglobin degradation. It also plays a key role in areas where erythropoiesis and active cell division occur. What is the structure of human serum transferrin? How does it look like? Human serum transferrin is a 79.6 kDa blood plasma glycoprotein made up of polypeptide chain containing 679 amino acids. It has a bilobal structure with two homologous lobes of almost equal size, namely N lobe, amino lobe and C lobe, which is also called the carboxy lobe, and these are joined together by linking peptides shown in the figure. N lobe consists of amino acids 1 to 331, while C lobe is formed by amino acids 339 to 679. Each lobe is further subdivided into certain domains, subdomains like N1, N2, C1 and C2, which are interconnected by two antiparallel beta strands that act as a hinge. Both the N lobes and the C lobes are capable of binding one iron atom with the help of 6 ligands. 4 ligands, which are aspartic acid, 2 tyrosines and a histidine that are provided by the protein, and 2 ligands provided by a synergistically bound carbonate anion described in the figure. Mode of action. How does this transferrin work? Iron enters into the cells by receptor mediated endocytosis in a form of monoferric and diferric transferrin shown in figure. So what happens is, first of all, the iron loaded transferrin binds to the receptors present on the outer surface of the plasma membrane. Subsequently, transferrin is internalized via invagination of clathrin coated pits followed by development of endocytic vessels. After that, the clathrin is removed with the help of uncoating enzymes and the vesicle then combines with the endosomes. The pH of the endosomes is reduced to 5.5 due to the activity of V-type ATPases in their membranes, which weakens the association between iron and transferrin. This would eventually facilitate the removal of iron from transferrin. Transferrin along with its receptor is then transported through the endocytic cycle back to the cell surface, which becomes ready for another round of iron uptake. So all this has been demonstrated in this diagram. Next, iron storage. So now that you have a fair idea about how iron is being transported from various parts of the body, we shall now get an idea about how iron is stored and precisely where. the iron that is not required in a functional capacity is typically stored in three major sites. Firstly, liver, secondly, bone marrow, and thirdly, spleen. It is the iron transport protein transferrin that brings iron to these sites, especially in the liver, where almost 60% of body's iron is stored, while the remaining 40% is found predominantly in the reticuloendothelial cells, also called as RE cells, within the liver, spleen, and bone marrow. Further, most of the iron present in the RE cells are obtained from the phagocytosis of red blood cells and the successive degradation of hemoglobin within the cells. So, the storage of iron primarily occurs in two forms. One, ferritin, and second, hemosiderin. Initially, iron is stored as a protein iron complex, ferritin. However, with time, the ferritin molecules aggregate to form clusters which are subsequently engulfed by lysosomes and consequently degraded. As a result of this process, hemosiderin, an amorphous aggregate of denatured protein, is formed. We shall now discuss about the individual roles of these two important iron storage complexes. First, ferritin. Ferritin is the principal storage form of iron in cells. It is water-soluble intracellular protein found in almost all living organisms, both prokaryotes as well as eukaryotes like mammals, plants, fungi, and bacteria, and synthesized in a variety of tissues, especially within the liver, bone marrow, spleen, and intestines. A typical ferritin molecule forms a hollow sphere which contains about 2000 iron atoms and may potentially hold up to 4500 iron atoms. The ferritin that has not been combined with iron is commonly termed as apoferritin. Let us get an idea about how ferritin looks like, that is the structure of ferritin. The structure of ferritin is of considerable heterogeneity and comprises primarily of two important components, a protein coat and an inorganic core suitably encapsulated within the coat. Figure shows the three-dimensional representation of ferritin, which clearly shows that this protein molecule is spherical in shape, containing iron atoms in the form of a mineral. So, you can clearly see in the diagram how this ferritin molecule is made up of three-dimensional ferritin molecule is spherical in shape and it is containing iron, which is primarily stored in the form of a mineral. The individual structures of the protein coat and the iron cores shall be discussed now. The protein coat is a highly conserved spherical assemblage made up of 24 polypeptide subunits. Each subunit consisting of about approximately 175 amino acids varying in molecular weight from 18 to 24 kilodaltons. Although the precise sequence of amino acid in the protein is not known, yet a similar type of amino acid sequence has been detected in ferritin isolated from either plants or animals. X-ray crystallography study reveals that each protein subunit is folded in the form of an ellipsoid as illustrated in figure, and these ellipsoidal units are further packed in a cubic symmetry with 4-fold, 3-fold and 2-fold axis. In the interior of the symmetrical protein coat, there exists a hollow sphere of approximately 100 angstrom diameter, which is surrounded by walls of approximately 10 angstrom thickness. At the intersection of these subunits, two separate channels are found which may be filled with varying amounts of solvent and iron. These channels have a four-fold and three-fold axis respectively and allow the movement of iron in and out of the protein, while the four-fold axis are lined with hydrophobic leucine residues. Three-fold axis channels are lined with hydrophilic aspartate, glutamate and serine residues. Next, we should get an idea about the iron cores. So, how do they look like and what do they comprise of? Chemically, iron in the ferritin core is stored in the form of a crystalline ferric oxyhydroxyphosphate complex that has the chemical formula FeOPO3H2.8FeOH thrice. The iron cores are unevenly arranged within the protein molecule, and it is only a fraction of Fe3 that binds directly to this protein. As revealed by X-ray crystallographic studies, iron is present as the mineral ferrihydrate 5Fe2O3.6H2O. Although the specific function of phosphate is not known, yet it is thought to be involved in binding of ferric oxide hydroxide units to each other as well as to the protein coat. Magnetic properties of ferritin. Now that we have an idea about the structure of the ferritin, we should know what are the properties of this molecule. Every single iron atom within the ferritin molecule contains 3 unpaired electrons. The spins from these thousands of electrons lock together under an external magnetic force via a quantum process known as exchange coupling. The core material is predominantly superparamagnetic in nature, as it acts as a solid piece of metal having single magnetic domains. Function of ferritin. Ferritin serves two major roles. Firstly, it provides a reserve of the element iron which can be drawn upon when needed for synthesizing important iron containing proteins such as hemoglobin, cytochromes and iron sulfur compounds. Secondly, it protects the cell from toxic effects of free iron which may otherwise catalyze the activation of dioxygen, leading to the formation of very reactive oxyradicals which may damage the body organs. How does ferritin store and release iron? So that's the next question. Ferritin stores and releases iron in a controlled fashion. Since ferritin has the shape of a hollow sphere, it stores iron in the form of iron 3, while releasing iron as and when required by the body. Iron 3 should be changed to iron 2 oxidation state. Once converted to iron 2 form, it leaves through the 3-fold and 4-fold channels in the spherical structure. Next, we move on to hemosiderin. What is hemosiderin? Hemosiderin is another iron storage complex that is found most commonly in macrophages and formed during breakdown of hemoglobin molecules. In contrast to ferritin, its molecular structure remains poorly defined. It appears to be a complex of ferritin, denatured ferritin and other material. The iron occurs within the deposits of hemosiderin, is poorly available to deliver iron when required. However, it is abundant in situations following hemorrhage that suggests that its formation may be related to phagocytosis of red blood cells and hemoglobin. Figure shows dark hemosiderin granules in Kupffer liver cells. The lysosomes assimilate large amounts of hemosiderin molecules in the cells that are overloaded with iron, which can be readily seen via Prussian blue staining as evident from the figure. Magnetic properties of hemosiderin. Hemosiderin is also super paramagnetic in nature, just like ferritin. However, in comparison to ferritin, hemosiderin's magnetic susceptibility effects are even more powerful owing to larger particle size. Next, we move on to iron disorders. Since you know iron is a very important element, so its deficiency or excess may lead to certain disorders which we shall discuss now. It is important for us to maintain an adequate supply of iron in the body in order to synthesize hemoglobin or other molecules that require iron for their proper functioning. Yet, because of everyday processes like urination, sweating, defecation, etc., our bodies keep continually losing iron in small quantities. For compensating these losses, one must consume approximately 18 milligram of iron every day. Also, there are instances wherein too much of iron is deposited in the body tissues that leads to a situation called iron overload. Following sections throw light on iron related disorders. Iron overload, that is also called as hemochromatosis. An iron overload disorder which makes the body absorb excessive amounts of iron from the food and drink we consume is often termed as hemochromatosis. Total body iron overload, or in other words hemochromatosis, occurs most often due to hereditary hemochromatosis or repeated transfusions in patients suffering from severe anemia. Hereditary hemochromatosis, an autosomal recessive disorder, is the most common of the two by far. Now, what is hereditary hemochromatosis and how does it occur? Hereditary hemochromatosis is a genetic disorder that reflects a fractional increase in dietary iron absorption. Under normal circumstances, humans extract iron from food through the intestines, and once iron is present in sufficient amount, the body reduces its absorption to avoid excessive accumulations. However, in case of people suffering from hereditary hemochromatosis, the body absorbs too much of iron, and in due course, that is over several years, this amount may increase up to 5 to 20 times. This excess iron in the body gets stored in the tissues of our major organs such as liver, heart, pancreas, which may eventually destroy these organs, causing severe chronic diseases such as cirrhosis, heart disease and diabetes. The gene responsible for hemochromatosis, which is HFE, identified on chromosome 6, encodes a protein that is homologous to class 1 HLA antigens. An alteration in this HFE protein basically involves the mutation of a cysteine to a leucine at position 282, in other words C282Y. People who inherit one copy of the mutant HFE gene are carriers who rarely develop iron overload and as such do not show any symptoms, since one correct copy regulates the iron absorption process. However, people with two inherited copies of mutant protein may develop this disease and consequently face a myriad of problems because of this. As shown in figure, if both the parents are carriers of the HFE gene, then their children may either be normal or carriers or may develop this disease hemochromatosis in case when they inherit both the copies of the genes, and this has been labeled in black. The most common symptoms of this inherited iron overload disorder include fatigue, weakness and lethargy, joint pains leading to osteoarthritis, etc. Next, we move on to transfusional iron overload. This iron overload disorder occurs in case of transfusion dependent patients like those suffering from thalassemia major, sickle cell disease, myelodysplastic syndrome, aplastic anemia, hemolytic anemia, refractory sideroblastic anemias and diamond blackfan anemia. Because of excess iron received during each transfusion and because there is no physiological means of excreting it, this element gradually accumulates in the organs of such patients, particularly in the liver. The assimilation of iron with repeated transfusions shows that heme iron atoms from the transfused red cells are retained after they have become senescent. The senescent red cells are ultimately destroyed by RE cells. Although the pattern of organ injury is similar in both transfusional iron overload as well as hereditary hemochromatosis, however, the distinguishing feature between the two is the occurrence of large iron deposits in the RE cells in case of the former. Lastly, we talk about iron deficiency anemia. Iron, as you know, is very important for the production of globular protein called hemoglobin, which is responsible for carrying oxygen from lungs to various body tissues for respiration to provide energy to power the functions of the organism. Hence, the deficiency of iron in the bloodstream leads to decrease in hemoglobin level, because of which organs will not get enough amount of oxygen, and such condition is termed as anemia. It occurs due to insufficient dietary intake of iron or iron loss from bleeding, either intestinal, uterine or urinary tract. Iron deficiency anemia can lead to fatigue, shortness of breath, chest pain and heart problems. So as shown in figure, you can clearly see how the normal and the anemic cells are different from each other. You clearly see that in anemic cells, the level of RBC blood count has gone down, and whereas in normal cells, you can see the normal amount of red blood cells. Summary. Now that we have studied each and every aspect of transferrin and ferritin, we shall recapitulate the points. Iron is an essential element for almost all living organisms since it plays a crucial role in a number of diverse cellular functions. Therefore, it becomes important to maintain a constant balance between iron uptake, transport, storage and utilization processes. Transferrins, which are widely distributed in the biological fluids of most vertebrates as well as invertebrates, are a group of non-heme iron binding glycoproteins that are capable of transporting iron from absorption centers to the sites of utilization, storage and hemoglobin degradation. The storage of iron primarily occurs in two forms. Firstly, ferritin and secondly, hemosiderin. Initially, iron is stored as a protein iron complex ferritin. However, with time, the ferritin molecules aggregate to form clusters which are subsequently engulfed by lysosomes and consequently degraded. As a result of this process, hemosiderin, an amorphous aggregate of denatured protein, is formed. An iron overload disorder which makes the body absorb excessive amounts of iron from the food and drink we consume is often termed as hemochromatosis. Total body iron overload occurs most often either due to hereditary hemochromatosis or repeated transfusion in patients suffering from severe anemia. The deficiency of iron in bloodstream leads to decrease in hemoglobin level, because of which the organs will not get enough amount of oxygen, and such a condition is called anemia.